turn a shower to cold right now. Step in, and something happens before you've decided anything consciously. Within 3 seconds, every cutaneous cold receptor in your skin fires simultaneously. Not one by one, all of them, across the entire body surface. Send signals up through the spinal cord to the brainstem at conduction velocities between 5 and 30 meters per second. The brainstem does not wait for a report from the prefrontal cortex. It does not consult your preference or your schedule. It issues a motor command before the cognitive brain has even processed the temperature. Your respiratory muscles contract sharply. Your heart rate spikes. Blood vessels in the skin and extremities constrict as the sympathetic nervous system redirects blood to the core. The gas that escapes is not a choice. It is a brainstem-level reflex with name, the cold shock response. And most people spend their entire lives never getting past it. The flinch happens between seconds 2 and 5. The hand reaches for the tap. The foot steps back. The decision to stop reads as self-preservation. And in one narrow sense, it is. The cold shock response evolved as a protective mechanism against cold water immersion, where the gasping reflex and cardiac acceleration serve to prepare the body for emergency swimming. The problem is that the nervous system cannot distinguish between a cold shower and a freezing river. The response fires identically for both. What the person stepping back at second 3 does not know is that the cold shock response peaks between 20 and 30 seconds of cold exposure, and then, if you stay, begins to subside. The cardiovascular response normalizes. The respiratory pattern shifts. A different branch of the autonomic nervous system starts to assert itself. The 90-second window is not arbitrary. It takes approximately 30 seconds for the cold shock response to peak and another 60 seconds for the physiological transition that follows to establish itself meaningfully. The biology is not compressed into the first gasp. It unfolds across a minute and a half, and almost nobody waits long enough to find out what happens after the first 30 seconds. Tibor Schellin at the University of Maastricht, who has spent 2 decades studying cold adaptation physiology, documented the cardiovascular timeline in controlled cold immersion studies. Heart rate during initial cold exposure spikes by 20 to 30 beats per minute within the first 30 seconds, then begins declining toward a new steady state by 60 to 90 seconds. Respiratory rate follows the same arc. The body is not stuck in the cold shock response indefinitely. The response has a natural trajectory, and the trajectory ends somewhere different from where it began. That ending point is the actual subject of this video. The first 30 seconds is the price of admission. The next 60 are what you paid for. If you're enjoying this story so far, don't forget to hit the like button and subscribe. Drop a comment telling me where in the world you're watching from. I love hearing from you. The skin contains 2 types of thermoreceptors relevant to cold. TRPM8 channels, which activate below 26 degrees Celsius and produce a sensation of coolness, and TRPA1 channels, which activate below approximately 17 degrees Celsius and produce the sensation of painful cold. A cold shower at 10 to 15 degrees activates both simultaneously across the full body surface. The signal volume is enormous. You have roughly 1.7 square meters of skin. Every square centimeter contains cold receptors. When cold water contacts all of it at once, the afferent signal reaching the brainstem through the spinothalamic tract is the largest cold input the nervous system receives under any normal daily circumstance. The brainstem processes this signal through the hypothalamus, which coordinates the autonomic response, and through the locus coeruleus, the brain's primary norepinephrine-producing nucleus, which we will return to in the next chapter. The immediate outputs are specific and sequential. The cardiovascular response comes first. The sympathetic nervous system increases heart rate and cardiac output within the first 5 to 10 seconds. Peripheral vasoconstriction follows as smooth muscle and skin arterioles contracts under norepinephrine released from sympathetic nerve terminals. Blood pressure rises transiently. The redistribution of blood from periphery to core is measurable within 15 to 20 seconds of exposure. The respiratory response runs in parallel. The cold shock gasp is driven by the sudden activation of cutaneous cold receptors, sending signals to respiratory control centers in the medulla. The initial gasp can be up to 2.5 liters of air involuntarily. Breathing rate increases from a typical resting 12 to 15 breaths per minute to 25 to 40 breaths per minute in the first 30 seconds. This hyperventilation lowers blood carbon dioxide, which causes cerebral vasoconstriction and contributes to the lightheadedness some people report during initial cold exposure. This is the moment when most people exit. What is actually happening at 30 seconds, when the cold shock response is at its peak, is a convergence of multiple sympathetic outputs that feel threatening because the nervous system is interpreting them as threat signals. The racing heart, the rapid breathing, the skin going cold, the slight dizziness. These are recognizable to the brain as markers of danger. They are also temporary. Mike Tipton at the University of Portsmouth, who has published extensively on cold water immersion physiology, documented the habituation of the cold shock response across repeated exposures. After 5 to 6 cold exposures over 2 to 3 weeks, the cardiovascular and respiratory peaks during the first 30 seconds are measurably reduced. The brainstem learns that this input, despite its intensity, is not followed by drowning. The peak dials down. The transition to the second phase becomes faster and cleaner. The biology is learnable. The first exposure is the hardest one. 30 seconds into cold exposure, the locus coeruleus, a small cluster of neurons in the brainstem, containing roughly 50,000 cells, has already begun releasing norepinephrine into the brain and the peripheral nervous system at a rate that no other daily circumstance produces. Norepinephrine is most commonly discussed in the context of stress. It is released during sympathetic activation alongside epinephrine, and its role in the fight-or-flight response is well documented. What is less discussed is its role in attention, mood, motivation, and the maintenance of prefrontal cortical function, and what happens when its levels drop chronically below the range the brain needs to operate cleanly. Andrew Huberman at Stanford has described the norepinephrine-dopamine relationship in the context of cold exposure specifically. Cold immersion at 14 degrees Celsius produces a 200 to 300% increase in norepinephrine, with the elevation persisting for 2 to 3 hours after the exposure ends. The data comes from a study by Tiina Mäkinen and colleagues at the University of Oulu in Finland, published in the European Journal of Applied Physiology in 2008. They exposed subjects to cold water at 14 degrees for 1 hour, a longer and colder exposure than most people will experience. But the directional relationship holds across shorter durations and warmer temperatures. 90 seconds at 15 degrees produces a meaningful norepinephrine elevation, measurably above baseline, persisting well beyond the exposure window. Why does this matter beyond the immediate feeling of alertness it produces? Norepinephrine in the prefrontal cortex modulates working memory, sustained attention, and cognitive flexibility through alpha, 2 adrenergic receptors on pyramidal neurons. When norepinephrine is in the optimal range, prefrontal function is sharp. When it drops, as it does with chronic stress, poor sleep, or sedentary behavior, the same cognitive capacities degrade. This is the mechanism behind the cognitive fog that follows weeks of accumulated sleep debt and chronic stress. The norepinephrine system has been running below its functional range. A 90-second cold exposure does not substitute for sleep or stress resolution, but the norepinephrine spike it produces is real. Its cognitive effects are real, and no supplement available over the counter comes close to replicating the magnitude of the release. Dopamine follows a parallel trajectory during cold exposure, with a separate study by Riedel and colleagues showing dopamine increases of approximately 250% above baseline during cold immersion. The combined norepinephrine-dopamine elevation produces the motivational clarity and sustained focus that cold exposure practitioners report consistently. This is not placebo. The receptors are responding to real molecular concentrations. The timing matters. Cold exposure first thing in the morning delivers the norepinephrine and dopamine spike during the window, when cortisol is naturally elevated, producing a compound arousal state that sets the neurochemical tone for the following 4 to 6 hours. Cold exposure in the evening produces the same neurochemical spike in the circadian phase, where arousal is less useful and may interfere with the melatonin production and sleep pressure that the evening requires. Morning cold, on both the neurochemical and circadian evidence, is the better placement. Something changes at approximately 30 seconds of cold exposure that most people who step back at second 5 will never experience. The cold shock response, having peaked, begins to decline. The cardiovascular and respiratory systems, having made their initial emergency adjustments, start receiving a different set of signals. And the vagus nerve, which, as the primary parasympathetic output to the heart, lungs, and abdominal organs, has been largely suppressed during the sympathetic dominance of the cold shock response, begins to reassert its influence. This vagal rebound is not unique to cold exposure. It follows any acute sympathetic activation that resolves without catastrophic outcome. The nervous system's design is oscillatory. Sympathetic activation followed by parasympathetic recovery, the balance shifting back toward rest once the threat signal subsides. What makes cold exposure different from most other acute stressors is the controllability and the repeatability of the stimulus. The brainstem integrates the sensory input from multiple channels simultaneously during the second phase of cold exposure. The skin is still cold. TRPM8 receptors are still firing, but the signal is now familiar enough that the emergency interpretation has begun to relax. The body temperature is stable. The cardiovascular system has made its initial adjustments. The carbon dioxide cleared by initial hyperventilation is beginning to rebuild as breathing rate normalizes. Arterial baroreceptors in the aortic arch and carotid sinus, which detect blood pressure, feed their signals upward through the vagus nerve to the nucleus tractus solitarius in the brainstem. As peripheral vasoconstriction redistributes blood to the core, central blood pressure rises modestly, which the baroreceptors detect, which triggers a vagal reflex that slows the heart rate. This baroreflex is a mechanical pathway through which cold exposure produces its most direct vagal activation. Stephen Porges, whose polyvagal theory described the autonomic nervous system as a three-tiered hierarchy rather than a simple two-branch system, would describe what happens at 30 seconds as a shift away from the sympathetic mobilization state and toward what he calls the ventral vagal state, the physiological condition associated with safety, social engagement, and efficient cardiac regulation. The measurable marker of this shift is heart rate variability, the variation between successive heartbeats, which reflects the balance of sympathetic and parasympathetic influence on the sinoatrial node, increases as vagal tone rises during the second phase of cold exposure. Resting HRV after regular cold exposure practice is measurably higher than baseline in multiple studies, including work from Lunt and colleagues showing significant HRV improvements after 4 weeks of twice-daily cold phase immersion. The practical implication is counterintuitive. Cold exposure, which begins as an acute sympathetic stressor, ends as a vagal training stimulus if held long enough. The nervous system that endures the first 30 seconds and stays for the next 60 gets a different biological outcome than the nervous system that registers the cold shock and exits. The exit feels like self-protection. Staying is the actual training. If you're hooked on this story, make sure to subscribe and turn on notifications so you never miss an upload. Your support means everything and helps me bring you more emotional stories like this one. When cold contacts the skin, the hypothalamus receives a temperature signal and issues a thermogenic response through the sympathetic nervous system. Norepinephrine is released at sympathetic nerve terminals in brown adipose tissue, the metabolically active fat depots found primarily in the supraclavicular region, around the kidneys, and along the spine, and the brown fat cells begin burning stored triglycerides to generate heat. This is thermogenesis, and it is qualitatively different from a heat generation of muscle shivering. Shivering is mechanical. Skeletal muscle contracts rapidly, generating heat as a byproduct of ADP consumption. Brown fat thermogenesis is chemical, uncoupling protein 1, abbreviated UCP1, embedded in the inner mitochondrial membrane of brown adipocytes, short-circuits the mitochondrial electron transport chain, diverting the proton gradient from ADP synthesis to direct heat production. The efficiency is remarkable. A single brown fat cell activated by norepinephrine can generate 300 times more heat per unit mass than a typical white fat cell metabolizing glucose. Brown adipose tissue, when fully activated, can account for up to 5% of total resting energy expenditure in cold-adapted adults, a metabolic contribution that is clinically meaningful across days and weeks. The discovery that adult humans retain significant brown adipose tissue was relatively recent. For decades, brown fat was considered a feature of newborn physiology present to help infants regulate body temperature before their thermoregulatory systems matured. PET-CT imaging studies published in the New England Journal of Medicine in 2009 by 3 independent research groups, Cypus and colleagues at Joslin Diabetes Center, Van Mark and Lichtenbelt and colleagues at Maastricht University, and Virtanen and colleagues at Turku University, simultaneously demonstrated that metabolically active brown fat is present in a significant proportion of healthy adults, concentrated in the neck and supraclavicular regions, and that its activation correlates inversely with BMI and age. The adult body retains thermogenic capacity. The capacity declines with age and with chronic warmth. Regular cold exposure reverses part of that decline. Wouter, Van Mark, and Lichtenbelt's laboratory at Maastricht has produced the most comprehensive human data on cold acclimation and brown fat. In a 10-day cold acclimation protocol, healthy male subjects spent 6 hours per day at 17 degrees Celsius. Cool but not extreme. After 10 days, shivering intensity had decreased while metabolic rate during cold exposure had increased. The body had shifted from an inefficient mechanical thermogenic response toward a more efficient brown fat-driven response. The sympathetic input to brown fat had strengthened. The UCP1 expression in supraclavicular fat biopsies had increased. 90 seconds of cold shower does not replicate 6 hours of cold acclimation per day. The brown fat activation from a brief cold shower is modest by comparison, but the norepinephrine signal reaching brown adipose tissue during cold exposure is real and measurable, and repeated daily stimulation produces cumulative adaptation in the tissue over weeks. The signal accumulates. The capacity builds. When cells experience rapid temperature drops, they produce a family of proteins called cold shock proteins, the most studied of which is RNA binding motif protein 3, abbreviated RBM3. RBM3 was identified as a cold-inducible protein in the late 1990s by Nishiyama and colleagues, but its functional significance remained unclear for a decade. The decisive work came from Banu and colleagues, and more recently from the laboratory of Banu at Oxford, who published findings in Nature in 2015 showing that RBM3 production during mild hypothermia prevented synapse loss in mouse models of neurodegeneration. The study used 2 mouse models, one of prion disease and one of Alzheimer's pathology, and found that mild cooling triggered RBM3 expression that preserved synaptic density and delayed neurological decline. Mice that could upregulate RBM3 survived significantly longer with preserved cognition. The mechanism is specific. During cold exposure, ribosomes, the cellular machinery for protein synthesis, preferentially translate RBM3 mRNA over most other transcripts. RBM3 protein then stabilizes other mRNA molecules that would otherwise be degraded, maintaining the cell capacity to produce proteins critical for synaptic maintenance. The net effect is a preservation of synaptic density under conditions that would otherwise trigger synapse retraction. Synaptic density is the structural substrate of memory and learning. The number and integrity of synaptic connections between neurons determines the richness and reliability of neural circuit function. Synapse loss is one of the earliest structural changes in Alzheimer's disease, preceding the appearance of amyloid plaques and tau tangles by years. Anything that slows synaptic loss is, mechanistically, a neuroprotective intervention. RBM3 appears to be one such mechanism, and its production requires temperature to drop, not dramatically, but measurably. The mild hypothermia that induces RBM3 in cell and animal studies is in a range of 32 to 35 degrees Celsius, achievable in peripheral tissues during whole-body cold exposure. The translation from mouse models to human biology is not yet complete. The Oxford group's findings generated significant attention in neuroscience, and human trials examining RBM3 responses to controlled cooling are ongoing. The direct evidence in humans for cold-induced RBM3 protection against neurodegeneration does not yet exist at the clinical trial level, but the mechanism is plausible, the animal data is strong, and the cellular biology is consistent across species. Cold shock proteins extend beyond RBM3. The broader cold shock protein family includes several RNA-binding proteins that collectively maintain cellular homeostasis during temperature stress. Their upregulation during cold exposure represents a cellular adaptation response analogous to the heat shock protein response that exercise and sauna exposure produce, but accessing a different set of molecular pathways. The heat shock proteins that exercise and sauna upregulate, particularly HSP70 and HSP90, are primarily chaperone proteins that prevent protein misfolding and facilitate protein repair. Cold shock proteins operate more on RNA stability and translational regulation. The two systems are complementary, which is why the combination of regular exercise and cold exposure, accessing both protein families, has attracted interest in longevity research. A single 90-second cold exposure produces measurable changes that last 2 to 3 hours. 30 days of daily 90-second cold exposures produce changes that persist between sessions. The distinction matters because most people who try cold exposure assess it by how they feel during the exposure or in the hour immediately after. The more meaningful question is what the baseline looks like on day 31, before the cold water even turns on. Vagal tone, measured by resting heart rate variability, improves across consistent cold exposure practice. The improvement reflects a structural change in autonomic nervous system balance. The parasympathetic system's capacity to modulate cardiac output has increased through repeated vagal activation during the second phase of each exposure. The nervous system that has been through the cold shock response 30 times has learned that the initial sympathetic spike is followed by safety, and the baroreflex that drives vagal rebound executes more efficiently with each repetition. Susanna Soberg, a Danish researcher who completed her PhD on cold water immersion and metabolism, has described the adaptation process in terms of what she calls training the thermoregulatory system, the body's capacity to regulate temperature becoming more efficient, requiring less cardiovascular disruption per unit of cold stimulus. Much as aerobic fitness develops through repeated cardiovascular challenge, the norepinephrine system adapts differently. Repeated cold exposure does not blunt the norepinephrine response the way repeated adrenaline exposure might blunt the cardiovascular response to a given stressor. The locus coeruleus continues to produce substantial norepinephrine elevations in cold-adapted individuals because the cold stimulus itself, unlike psychological stressors, does not habituate fully at the neurochemical level even as the cardiovascular shock response diminishes. The body gets better at managing the cold. The brain keeps responding to it. Brown adipose tissue, as Van Mark and Lichtenbeld's cold acclimation data showed, increases in metabolic activity and in UCP1 expression over weeks of regular cold exposure. The increase is not dramatic from brief shower duration exposures, but it is present and cumulative. Brown fat that receives daily norepinephrine stimulation maintains its thermogenic capacity better than brown fat in thermally comfortable environments. The tissue that is never challenged adapts toward inactivity. The psychological changes are harder to measure, but consistently reported. People who complete 30 days of daily cold exposure, without exception, describe a change in their relationship to discomfort more broadly. The specific change is not vague resilience or motivational improvement. It is a recognizable experience of having entered an acutely unpleasant state deliberately, survived the peak of it at 30 seconds, and arrived somewhere different at 90 seconds, 30 times in a row. The nervous system that has this pattern encoded is different from the nervous system that flinched away at second 3 every time. It has a reference point. It knows the arc. The 30-second peak is identifiable. It is survivable, and what comes after is known. The translation of this into non-cold context, the difficult conversation, the challenging task, the moment of acute social discomfort, is not metaphorical. The same prefrontal cortical circuits that override the cold shock flinch are the circuits involved in voluntary delay of aversive experiences generally. Alia Crum at Stanford has studied the relationship between mindset and physiological stress response, and her work suggests that the framing of a stressor, whether it's perceived as threatening or as challenging, changes the hormonal profile the body produces in response. Cold exposure, practiced deliberately with attention to the arc of the experience rather than the escape from it, trains the framing alongside the physiology. The experience is unambiguously aversive at second 3. The body produces a threat response. But the person who has done this 30 times knows the threat is temporary, and that knowledge is encoded in prefrontal circuits that modulate the amygdala's initial alarm. The person who has never stayed past second 5 does not have this encoding. They have only the alarm. What changes after 30 days is not that cold stops being cold. The TRPM8 receptors fire at 15 degrees, whether it's your first shower or your 100th. The cardiovascular spike happens. The gasp happens. What changes is the nervous system's capacity to observe the spike without identifying as evidence that something is going wrong, and to stay in the experience long enough of the biology that follows the first 30 seconds to do its work. The 90 seconds is not the difficulty. The 90 seconds is the treatment. The first 30 seconds is what you have to pass through to reach it. Stand in the cold for 90 seconds. Every morning, the norepinephrine will spike on day 1 and on day 30. The vagal pivot will happen on day 1 and on day 30. The brown fat will receive its activation signal on day 1 and on day 30. What changes across those 30 days is the fraction of a 90 seconds that feels like an emergency and the fraction that feels like something else entirely. By day 30, most of the 90 seconds feels like the second thing. Thank you so much for watching until the end. If you loved this story, check out the other videos on your screen now. I think you'll really enjoy them. And don't forget to subscribe if you haven't already. See you in the next story.